So in this section, I will introduce you the knowledge of phase contrast microscope. It's very important because you will use it in your exercise 3. So at the very beginning, let's see what is contrast. So contrast describes our ability to distinguish two objects based on their relative intensities. So in order to let our eyes sensitive to the image, we need to have a higher contrast. So how to enhance the contrast of the sample? One of the methods we mentioned before, we could stain the sample. So as you see here, after staining, because the sample will have these different colors, and also the color shows in different structure, it could be seen or recognized with the bright field microscope. And also there's other method could increase the contrast. So for each method or technology, it leads to one type of light microscope. So in this section, we will see the phase contrast microscope. So just at the very beginning, I would like to introduce you a little bit light, the physics of light. So here shows you the light wave, and you see here between the two high peak, it's the wavelength. And for the distance from the high peak to the baseline, it shows you the amplitude. So the wavelength actually gave you the difference. The difference on wavelength gives you different color. So as you already know that for the purple side or the blue side, they have short wavelengths. And for the red side, it has larger or longer wavelengths. The amplitude of the light, it gave you the intensity. So the light is strong or not, it depends on the amplitude. So your naked eye is actually quite sensitive to the changes of wavelengths and the amplitude. So when the color shifts, all the intensity of the cells, of the light shifted, your eyes could easily recognize. For example, here shows you the amplitude specimen. So here's the sample. When the light passes through the sample, your sample, you can see that although the wavelength didn't really change, but the amplitude or the light intensity changed. So at this time, it's the theory under the bright field microscope. Your eyes could easily recognize the difference. And the other type of specimen, they didn't really change the intensity of the light. But if you see here, when the light goes through this specimen, the wave changes a little bit smaller when they inside the specimen. But when they get out of the specimen, there's no changes on the wavelength. But since it goes faster here, the phase, we call it phase changes. So as you can see here, let's see here at the time 0. So without specimen, at time 0, there's a high peak of the light. But when you see here, it's moved a little bit back. So we call it 90-degree shift, phase shift. The problem is with this kind of sample, usually it's a transparent, thin sample. Your eyes couldn't really see the differences. That's why in the previous slides, you couldn't distinguish the bacterial cells from the background. And here is another samples. It's changed on two sides. So not only the phase shift, but also the intensity changes. So at that point, your eyes quite easy to recognize the differences. So what is the principle of the phase contrast microscope? It is actually to try to convert the differences in phase into the differences in the amplitude of the light. So here shows you the phase contrast microscope, and here's the light path of this type of microscope. So I have to say that the phase contrast microscope is the first microscopic method that allows the observation of the living cells, include the bacterial swimming, the bacterial cell division. And it is also the most used microscope in the biological labs, which you will also use in the exercise 3. So let's go back to the light path. So as you remember, in the bright field microscope, the light goes through from the light sources. And when they go, before they just enter the condenser lenses, there's an extra material or sense, we call it a phase ring, could filter the cells and try to have only cells with the direction, the same direction to go through this material. And this, this is not between the two lines. Let's imagine if you look through from here, you can see the light only happens as a circle. And when they go to the condenser lens, this circular light will be focused at the place of a specimen, and you will see the little spot. And when the light goes through the specimen, it will be detected by the objectives, the lens in the objectives, and try to, when they go through it, some light go directly, without a problem, go directly to the next specific material of phase microscope. We call it a phase ring, phase plate. So in the phase plate, you can see a ring here. They use different material here and the ring and the outside. So when the light goes through the center, the ring, the light will be changed out of 180 degree out of phase. But when the light, the underivative light goes through the center material or the side material, it will not change, so it's only 90 degree phase changes. And when the different light try to focus in your eye, which is not shown here, it happens the light interference. So the wave will be a little bit changed. The wave, the phase changes, shifts will be changed to the amplitude differences because of light interference. Oh. So at the beginning of this video, it shows the principle of a dark field, which shares some common principle as the phase contrast. So what you can see here is the specimen. This is the image plate, which is magnificent to here. So what you can see, this is the specimen insert into the light path, and it's focused on the imaging plate, and you can see the image here. So this happens also for the phase contrast. So try to focus on the changes on the light and also what happened on the light plate, on the imaging plate. So now it goes to the contrast. And here shows you the black one is the phase ring. And now they put the sample, and you can see the light path just be changed by this sample. And now they add this phase plate, and you can see the wavelength, the light is a little bit retarded by this material, and they try to have this interference of the two light, and the image of the specimen just be increased, the contrast is increased. Well, if you want to know the details, or if you want to re-see this video, just go back to the PowerPoint, the slides, and try to click it again. Mm-hmm. So let's continue with let's continue with the phase contrast microscope. So here I show you this is the first Nobel Prize that mentioned in this lecture, because the phase contrast microscope, it was invented by Fritz Ziernik in 1934. So he actually won the Nobel Prize in Physics in 1953 because of developing phase contrast microscope. So if you are interested in the what happened, you can go to the website and try to find the lecture he gave in the Nobel Prize. Mm-hmm. And now let's go back to the theory of what happened in the phase contrast microscope. So as you already see in the video and the previous slides, that there's two specific components in this phase contrast. And the first one, we called it phase ring. In some materials, they also called it annular ring. And it is, the function of this is restricts angles of illumination. So diffracted and undifracted light can be selectively modeled at phase ring plate. So this is the phase ring plate. Well, the material of the phase ring plate is a little bit different. So there's three parts. In the inner part and the outer part, they made of some portions they coated with light retarding material. So when the light goes through this material, it will be retarded about 1/4 of the wavelength, so 90 degrees. But when the light goes through the center material, the material of the center, it didn't give any changes to this to the light go through here. So they will have different phase shift of the two light, and then it's more easier when they interfere with each other. It will be more easier to be recognized because of the changes in the intensity. Mm-hmm. And so what exactly happens when the light passes through the specimen? So here shows you two things actually happened. We go here. So when the light goes through the slides and your sample, there's different things happens. So when there's no sample, only the slides, we can see that because it's glass and very few changes on the face, we can see that the light goes directly here without any changes. We call it kind of background light. But when there's a very thin sample here, the face will be shifted or changed a little bit when they go through the little things, the very thin samples. You can see that there's a very minimal, very few changes on the phase shift. We call it delta 1. Normally, the size for the changes is 1/4 of wavelength, so it's 90 degrees. And when the sample is really thick, for example, here, when they go to the thickest portions, it will be changed a lot. Not only on the, as I showed you in the previous slides, not only in the intensity or the amplitude, but also in this big shift on the phase changes. So this could be easily, if it happens like interference with some light without changes, it could be easily recognized by yourself, by your eyes. But when they go to the very thin transparent sample, this meaning the shift here is too small, and your eyes couldn't really recognize. So the second thing is, on the surface of your specimen is normally uneven. So it's not a flat homogeneous surface. Normally they have different angle of the surface. Then what's happened to the light when they go through here? The refraction happens on the surface, but it also depends on the angle of the surface. So it can go in different direction. So when the light goes, for example, there's a refraction light, go into the center of the phase ring, the circle of the phase ring. As you remember from the previous slides, there's no retard, so there's no changes on the phase. But when they go through this material, the light will be shaped a little bit, and they will become bright. So basically, the contrast will be increased between the two different places, and your eyes could easily recognize the differences. Well, in these slides, you can see the same thing observed under the bright field microscope and the phase contrast microscope. So what I want to show you here, actually, you can see this is a bacterial. You can see they are black under this phase contrast microscope. But when you try to find this kind of bacteria here under the bright field microscope, it's hardly be seen. So probably here and here, but you couldn't really see the shapes or more further. And so the important thing when you use a phase contrast microscope is to align the two components to be well aligned. So I will show you what is really located inside. So here I will show you with, I need to take out the condenser. So when you try to work with your exercise, don't do that by yourself. If you have any problem or you want to observe it by yourself, try to ask one of the TA or teachers. Sorry. Okay. It's okay? Yeah. Sorry, just hold it. Well, so here shows you now you can see the lens or the lens, or there's no any modification here. It's 0. So this will be used for the bright field microscope. But when you try to turn here, here you can see a little word, right, phase 1. And you can see the little ring on the phase 1. So it is the phase ring. Can you see it?
Yes.
Yes. So when you turn a little bit, they have 3 different sides. Now it goes to the phase 2. And phase 2 show a little bit larger. You can see the ring. Mm-hmm. And they also have another size, phase 3, the largest one. When you continue to turn this, you can also see there's a board for the dark field. So this is all that's in this condenser. But remember, don't remove it by yourself. And after I will show you, so we need to put it back. Okay, to work carefully. Oh, yes. You're right. Yes. And then we will use this small screw to tighten it. So the important thing is when you try to use, for example, the objectives. For example, this one, the 10 times objectives, you can see that there's a marker as a pH1. It means phase 1. Then you need to allocate the plate here as phase 1. So they could be the ring here will be faced into the ring inside the objectives. Then you will get the best resolution and of your image. Mm-hmm. So here shows you details that if it's arranged very well, you can see that the light rain here goes through this plate. But when it's not well arranged, you can see there's only a light spot and all the other parts are dark. Therefore, they couldn't really be focused on your eye and you couldn't see anything. So this is very important part. And this alignment will be done by the teachers before this your exercises. So you don't need to do anything here, but you need to know this theory. Well, if you really want to try by yourself, there's a little online games. You can just click here. There's a link here. Go to the website and try to remove and adjust the rings. And if you have time, of course. Mm-hmm. And another important thing is when you try to observe the cells, it's very important to let the condenser or the iris diaphragm in the center, so the light can easily go to the right place and you can see the sample. So I will also show you a little bit how to adjust it, especially when you remove this condenser and try to rearrange, try to input back again, then you need to fix everything. So first, again, we need to let's increase a little bit the light. Then it's more easier for you to see the light happens. So increase a bit the light and turn the iris to the. So this is the largest. Just 10 counterclockwise to the minimum size, and use the 4 times objectives. And there's a small screw here that you can move the condenser up and down. So let's try to move up. And also the important thing is when you try to look through the eyepieces, which I didn't mention previously, you really need to adjust the two eyepieces according to the distance of your two areas, pupils. Yes, the eyes. So try to turn a little bit the distance. So what I did is actually I looked through the two pieces at the same time and try to turn the two circle fields into one. But you also have other methods. For example, there's numbers here shows you the distance, and you can ask your friend try to measure the distance between your two eyes and fix it, just set up here. And another thing is try to ask one of your teammates, try to observe if the light goes directly to both of your pupils. Yeah. Well, another thing is you can use paper. You mark the light. You mark your eyes distance maybe, and try to thin it. If we just turn a little bit. You can see it's not really focused on the eyes. So what we want both of them focus on each of your eyes, pupils. And you can see the sharpness of the two spots are different for different eyes. So it's also because the eyes for your both of eyes, your eyes might have different lengths, or because, for example, I have the glasses, I have -3 and -4 for one of them. And then you need to adjust when you try to close one of your eye and make a focus here, and then try to close another one and open another one, try to look through and focus for each. And at last, try to focus on both. And turn a little bit here, but not too much. When you have problems, just ask one of the TA or the teachers. Well, let's go back to the century erasers, the condenser. So here, let's start again. First, open the light to maybe the highest, and then minimum here to have the minimum size of the iris. And then try to use this screw here to move up, and then look through the eyepieces. So mine is 59.5, so I just, now it goes. Move further up until you see there's a sharp edge of the light. You can see there's a very sharp pattern here. Yes. And then you increase. So if it's not in the center, if the light spot is not in the center, then try to use the two screw here to move either X in the one direction and the other one is in the other direction. Try to move the spot as exactly the center. So how do you know it's exactly the center or not? Try to, when it's become sharp, so I need to reduce a little bit the light. Try to increase until the edge of the pattern just as it touch the circle, the circle of the light. And if it's not if it's not in the center, you will see that one side might be covered by the block, and the other one you still have this distance. So try to remove a little bit here, and try to let the condenser at the center, and then you will have the best image and best light light pass.
Which screws was it?
It is the screws here.
Yes.
So this one. So this one is when you try to remove the condenser, the small one. And the two small here is this. Oh, I didn't show you here. Here, actually, the things that's linked to some plate here. Don't do that. Don't touch anything here when you try to have the best image of your sample. So if you have any problem, ask one of the TA or the teacher. Yeah, we will adjust it to you or help you to adjust it. Okay. Mm-hmm. So. Well, of course, for a phase contrast microscope, there's both advantage and disadvantage. So I will not read through all this. When you have time after this, when you want to know more details, try to read the 2 slides at the end of this this lecture. If you have any questions, just ask me or one of the teachers.